Welcome to the world of Gametogenesis. My name is Dr. Byson Em. Let's get into this class. What is gametogenesis? Gametogenesis is simply the process by which male and female sex cells are produced through the process of meiosis. Now, the male sex cells are called spermatozoa. They're commonly known as sperms. The female sex cells are referred to as the ovum, sometimes called the egg. Gametogenesis in females is called oogenesis and takes place in the female gonads. The female gonads are known as the ovaries. Gametogenesis in males is called spermatogenesis and takes place in the seminiferous tubules of the testis. Now, let's go deep and look at oogenesis. Oogenesis is the process by which female sex cells are formed through the process of meiosis. So the process by which oogonia differentiates into mature oocytes in the ovaries. Oogenesis also refers to the sequence of events by which the primordial germ cells are transformed into mature oocytes. Now, what you need to know is that the primordial germ cells come from the endoderm layer of the primitive yolk sac. Oogenesis starts before birth. Now, what happens? The primordial germ cells migrate from the developing, they migrate into the developing ovaries at week 4 of development from the primitive yolk sac and differentiate into oogonia through the process of mitosis. The oogonia will be surrounded by follicular cells, and by the 3rd month of development, the oogonia will undergo mitosis and will differentiate into the primary oocytes. Now, this is a schematic diagram showing the process of how oogenesis takes place. So you have the germinal cell here. It is diploid, meaning it has 46 chromosomes or 23 pairs of chromosomes. This germinal cell here is what we are calling the oogonia. Remember, the oogonia comes from the primordial germ cells. All right, so the oogonia, which is the germinal cells, will undergo mitosis and will develop into what we call the primary oocyte. The primary oocyte is also diploid. The primary oocyte would then enter meiosis 1. And when they enter meiosis 1, they will produce one secondary oocyte, which is a haploid, and the first polar body. The polar body is a non-functional cell because it doesn't have cytoplasm. It has little to no cytoplasm. So this is non-functional. The secondary oocyte is the functional oocyte. Just remember that when the primary oocyte, which is diploid, enters meiosis 1, it will produce a secondary oocyte, which is a haploid. Secondary oocyte will then enter meiosis 2. When it enters meiosis 2, this secondary oocyte will produce an ovum, which is a haploid, meaning it has 23 chromosomes and a second polar body. Polar bodies are non-functional. At the same time that the secondary oocyte was entering meiosis 2, the first polar body will also enter meiosis 2 and will produce 2 polar bodies. So in total, at the end of oogenesis, 3 polar bodies are formed. Now, the right side here shows how it happens in the ovary. So this is the ovary. These are the primary oocytes. These are the secondary follicles. And this is the Graafian follicle, which contains the secondary oocyte just before ovulation. And what is happening here is ovulation, meaning that the ovum is being released from the ovary into the fallopian tubes. All right. So this is a summary as well. Primordial oogonium, which is diploid, develops into a primary oocyte. It is arrested in prophase 1. So the arrest in prophase 1 is at the diplotin stage of prophase 1. Okay, and then this primary oocyte after birth will enter meiosis 2 and will develop into secondary oocyte. This secondary oocyte that develops is a haploid cell. Okay, is a haploid cell, and this is what is going to get fertilized. So that is the process of oogenesis. It's very simple and straightforward. Okay, same diagram referring to what we've been talking about. Oogonia enters the primary follicle through mitosis. Primary oocyte. The primary oocyte enters meiosis 1, develops into a secondary oocyte. Secondary oocyte enters meiosis 2 and provides an oocyte. An oocyte is an immature ovum. It differentiates and develops an ovum, which is mature oocyte. Always remember that at the end of oogenesis, 3 cell bodies are produced. Okay. So this is an ovulated oocyte. This is a mature ovulated oocyte. This is the structure. Everyone is required to know this. So these outer cells are what we call corona radiata. And then this is the zona pellucida, the vertilla in space. This is the germinospore and the germinosac and the ooplasm. So that was all about oogenesis. Now let's talk about spermatogenesis. This is the process by which spermatozoa, commonly referred to as sperms, are produced through the process of meiosis in the seminiferous tubules of the testes. Spermatogenesis begins at puberty. Okay. So at birth, germ cells in the males can be recognized in the sex cords of the testes as large pale surrounded by supporting cells. Supporting cells that support the germ cells of the germ cells of the testes are known as the tentacular cells or the Sertoli cells. Shortly before puberty, the sex cords acquire a lumen and become the seminiferous tubules. So now, this is the part of the seminiferous tubule. This is a cross section, and this is the part of the seminiferous tubule. This is the diagram they bring in tests increases. So everyone is supposed to know this. So this is a spermatogonia. It's a germ cell. The spermatogonia will undergo mitosis and will produce a primary spermatocyte. The primary spermatocyte will enter meiosis 1 and produce a secondary spermatocyte. Secondary spermatocyte will undergo meiosis 2 and produce a spermatid. A spermatid is an immature sperm cell. So the spermatid will undergo through a process called spermogenesis and will mature into a spermatid. Okay, so let's look at the diagram showing that. So this is a spermatogonia. It will undergo mitosis and will produce a primary spermatocyte. Now, as you can see, spermatogonia is diploid. Primary spermatocyte is diploid. The primary spermatocyte will enter meiosis 1 and produce a secondary spermatocyte. The secondary spermatocyte is haploid. So each of the 2 spermatocytes, secondary spermatocytes that are produced, will enter meiosis 2 and will produce 2 spermatids each. So 4 spermatids are produced from 1 spermatogonia. These spermatids will undergo a maturation process known as spermogenesis and will mature into spermatozoa. That is how spermatogenesis happens. Okay, so this is just an illustration of how spermatogenesis happens and how oogenesis happens. All right, and this here is showing how the fusion of the sperm and the ovum takes place, the process of fertilization. If you haven't watched our video on fertilization, just the link will be in our bio. Go and watch it. This is a mature sperm cell that is produced, which has the head. The head has got an acrosome. This acrosome has got enzymes that are used in fertilization. The neck, the mixed piece, which contains mitochondria, which produces the energy for propulsion, the movement of the sperm. The structure that the sperm uses to swim or to move is the tail, also known as the flagellum. So that is how spermatogenesis happens, and the resultant structure here, which is the sperm cell. Okay, now let's look at the normal semen analysis according to the WHO 2010 guidelines. The sperm volume is supposed to be greater than 1.5 mils. And the sperm concentration is supposed to be greater than 15 million mil. Total sperm count is supposed to be greater than 39 million. Sperm progressive motility is supposed to be 32% of the sperms that are released are supposed to be motile, supposed to be moving. Sperm morphology, normal, is supposed to be greater than 4%. The sperm DNA fragmentation should be less than 30%. Non-sperm cells should be less than 1 million per mil. So this is the normal guidelines of the sperm according to WHO. So know this. Sometimes it comes in exams and you're supposed to know these values. Very important. Abnormalities of the semen analysis. Okay, so if you have sperm concentration that is less than or equal to 15 million sperms per mil, that is known as oligozoospermia. Oligozoospermia. If the sperm progressive motility is less than 32%, that's called asthenozoospermia. If the sperm normal morphology is less than 4%, that is called keratozoospermia. All of the above three parameters, if all of the above three parameters exist, meaning if there are these three anomalies in one individual, that is called oligoasthenokeratozoospermia. Azoospermia is where no sperm is detected in the semen. They ejaculate. If there are no sperms, that is known as azoospermia. Normospermia is where there are normal sperm parameters. What we talked about here, this is called normospermia. Right, that is the end of our presentations. I hope you learned. Watch out for our next video. Dr. Byson Em. Don't forget to like, subscribe. See you in the next video.